All right, we are now going to go over as much as we can of muscle. Okay, so muscle tissue, I should say. All the main muscles we just do in lab, because it would be me standing here just like with bones, this muscle and that muscle. We're going to do them in lab. Okay, we're going to give you a worksheet. We're going to do a little bit of work at home, then we'll go over all the muscles in lab with you. Okay, so this is the next lecture, which will be muscle tissue. Is not the final stretch. I used to put this at the end. Sorry, I should change it. You're like, oh man. Mark my words though, the next 3 weeks will drag, maybe 4, and then you're going to blink and you'll be done. That's how it goes in the spring. You know, it feels like torture right now for some reason. I think it's just the weather. It does, I'm telling you. You get through the next 3 or 4 weeks, you know, just be extra good to yourselves, and then you blink and you'll be done. So, all right, so overview of muscle tissues. Do we remember the 3 major types of muscle tissues? Skeletal, cardiac, and smooth. So, skeletal is attached to the skeleton. Part is cardiac. Where's smooth? Yep, those hollow organs, uterus, blood vessels, GI tract, okay? So we'll look at them briefly microscopically in lab. They have different microscopic attributes. I don't know if I get too much into that here. I don't think so. Myo, Mys, Sarco are all prefixes for muscle. So you might hear those different prefixes depending, because we don't like to make it easy, you know, kind of keep it complicated. Why not, right? All right. Now, myofilaments are the little filaments that we will study in those muscles, and I'll talk about those as we come up to them. All right. If you remember looking at skeletal muscle under the scopes, you get the long muscle cell, and then inside that you have those fibers, and then you also have those transverse light, right, lines that we went over. So we'll look at those in lab. But the myofilaments are little fibers that help those muscles to contract. Now, I want you to sort of get one premise into your head really quickly. Muscles, when they're activated, shorten. Muscles, when they're activated, shorten. You can't activate them and lengthen them. Are you with me? How do you lengthen a muscle? Think about exercise. How do you lengthen a muscle? You stretch it. So I can't like think really hard and lengthen a muscle. You can't do that. And it's the way those little myofilaments cross over each other that make that happen. I'll show you in a little bit, okay? So, skeletal muscle has those striations. Do you remember what does that make sense? Okay. Remember what those striations are? The light and dark alternating lines that go transversely over that muscle fiber. Those striations, it's where those little myofilaments overlap. That's why you get those. So we have 2 different myofilaments that overlap, and where they overlap would be darker, and where they don't would be lighter, because there's less of them. It's basically what happens. Now, it's voluntary, obviously, because we can make ourselves move. And it tires easily, so that means it makes a lot of and uses a lot of what? ATP. If you remember way back from maybe the 3rd week of class in the fall, we need what to make the most energy? Glucose. Muscles, skeletal muscles specifically, and the liver store glucose as what? Glycogen. You store glycogen in skeletal muscles and liver primarily. Liver is where we store it to share with everybody else. So if you go a full day without eating, right, you need more glucose. We can tap the liver to that stored glycogen and cleave off glucoses, okay? We also store it in the skeletal muscles. The thing about the skeletal muscles is they tire easily, so guess what? Do they share their glycogen? No, they're very selfish, skeletal muscles. and they'll keep it to themselves so they can create a lot of ATP. And look at how much I've been moving just this morning, right? You need a lot of energy to keep those skeletal muscles going. By the way, this picture doesn't look anything like our slides, so don't look at this and think, oh my gosh, right? We'll look at ones that look better in a little bit. All right, cardiac, found in the heart, right? Now, cardiac is striated but not voluntary. And let me turn on the lights really quickly just to give you a little refresher about those microscopic attributes. So skeletal has a long cell and it has many what? Nuclei. And those striations are going to be the light and dark lines that are the crossing over of the actin and myosin, or those myofilaments, which we'll get into a little bit. Now, cardiac looks different in that cardiac cells are what shaped? Y shape. They're also striated, so cardiac cells are Y shape. They're mononucleate, which means one nucleus, and they're striated throughout the whole cell. Now, why do we want to have them Y-shaped? What do you want to do? You're sending, it's like gossip. One person talks to two, right? Two talk to four, it's exponential. Because you want to send that, I'm not saying to go gossip, you realize I'm just joking. I'm just trying to give you an analogy. I hate gossip. Okay? You want to send what quickly? Signals through the heart. So if one, it's exponential, you see. One talks to two, two, four, etc., etc. Are you with me? Okay. So we have that Y-shaped cell to be able to do that. Okay. Now, smooth, and it's not voluntary, cardiac. Okay. Can you get stressed and affect your heart? Absolutely. But for the most part, the heart is dealt with involuntarily. Okay. Smooth muscle found in hollow organs. I give you a list. Okay. Are those cells striated? Nope. They're long and spindle shaped with one nucleus in the center. You can see this in this picture here. And they're all kind of smushed together. And do you remember looking at them in lab? And I said it's almost like you take a dried paintbrush and you create like a half circle. Because we have, I think, part of the intestines that we look at the smooth muscle in. They're those long spindle-shaped ones. They're not as obvious, I think, on the slide as they are in this picture, okay, of that smooth muscle. Okay, so not striated, involuntary, found in hollow organs. Yes? Okay, great. I think this all makes sense. Okay, excitability will happen with the action potentials. We also use calcium, which I'll talk to you about how calcium comes into play. They can shorten, they can release because they can be stretched, but remember we can't forcibly lengthen them. And then elasticity, we have a little bit of recoil with our muscles. So you can probably see that when you stretch. How long do you have to stretch to really lengthen your muscles? A pretty long time, because they do have an elasticity where they do want to recoil back to their original shape. Okay. Now, movement's pretty obvious. I think we sort of get that. Posture. This is something to start thinking about. You have something called tonicity in muscles. It's in your postural muscles. It's also like in your GI tract. And what that tonicity means is there is a slight contraction to keep that muscle ready to work, but in the GI tract. In your postural muscles, that tenacity helps to keep you upright. Your posture muscles keep a slight amount of contraction at all times when you're upright to keep you upright, right? Think about it. If you weren't constantly micromanaging your back muscles and your neck muscles, you just fall over. It's actually a reflex in us to keep the dens, which is on which cervical vertebra? Axis. Number 2, you got it, right? Over your sacrum. If you walk around like, I don't advise it, but if you walk around like this in the hallway, is that gonna feel normal? No. Your body wants to keep the dens over your second sacral tuberosity, or tubercle I should say, and keep you upright. That keeps you balanced, but it also keeps your eyes what? With the horizon, parallel with the horizon, so you can keep binocular vision. Are you with me? Okay, so those postural muscles are constantly keeping us upright so we can keep those 2 abilities. Muscles help to stabilize joints. Muscles also create heat. This is a huge thing that our muscles do for us. Huge thing. It creates the majority of the heat that we have in our body. That's why if you get really cold, you start to shiver, because your muscles are trying to generate heat to warm you up. Okay? So nerve and blood supply, each muscle has 1 nerve and 1 artery. Once it enters, it does what? Splits and splits and splits and splits to get through that whole muscle. Different muscles will have different amounts of splits. Okay, finer muscles will have more. Bigger muscles like our back muscles will have less. Okay, but it'll split down to those different muscle fibers. Enters the center of the muscle. Each muscle fiber has a nerve ending. Needs lots of energy, which we spoke about. There's a cool picture up there of the nerve breaking down. Okay. Connective tissue sheets. So if you look at this, maybe if I turn off the light, it'll be a little bit easier to see. All right. So we have this epimysium around the whole, so this is one muscle, okay? The epimysium is around the whole thing. The paramysium is these little subdivisions. And then the endomysium is actually around one muscle fiber or cell, all right? And if you can imagine this packaging and this packaging and this packaging all comes back and fuses into that tendon. Now, why would we want to do that, do you think? Why would we want to take all those connective tissues and fuse them into that common tendon? Nobody?
Extra support.
Extra support and even pull on that muscle, right? If you're taking all of those wrappings and attaching them to a common insertion or origin, it'll pull on all those muscle fibers, or I should say all those muscle fibers will pull on it evenly. Are you with me? If you had a whole bunch of connective tissue going one way and not the other way, all these guys would work and these guys would be left out. So it provides nice even pull across that muscle, right? Even if you do damage a little bit of this tendon, you know, you still have other ones that can pull on that tendon and help to move it. Does that make sense? Okay, so all of these, the endomysium, paramysium, the paramysium just makes fascicles, so each one of these subunits is a fascicle, okay, and the epimysium, that's their whole job, is to provide a nice even pull across that muscle, okay, and subdivision. So attachments, insertion, origin, we went over this, okay, in the last lecture in joints, so I'm not going to go over it again. If you have any questions on it, come see me. Now, the attachments can go into a tendon, which is a very narrow connection of connective tissue from the muscle to the bone, or it can go to what's called an aponeurosis, And aponeurosis is simply a wider connection of dense irregular connective tissue. Okay, it can go into a tendon or what's called an aponeurosis, okay, which is a wider connection. Like we have one at the bottom of our foot that goes across the whole width of our foot, for example, okay? Now, what they do, both of them, the tendon and the aponeurosis, is it'll blend into the periosteum of the bone. Back to this picture, okay? They'll come up and they'll blend into this periosteum of the bone to give a nice connection that's strong, okay? Any questions so far? Are we good? All right, we're almost there. Take a deep breath. Okay. So the way that it's structured, if you could picture back to that previous picture where we had the endomysium, we had one fiber there, that's one muscle cell, okay? That muscle cell is long and tubular, multinucleate, just like I drew on the board. And inside of it, we have many, many myofilaments, which are called actin and myosin. Does that sound familiar at all? No? Okay, they're these teeny tiny fibers which help to create the movement in that muscle cell itself. And of course, instead of calling it by regular names, we have all these funky, names for it. Okay, so here's our muscle cell, we're talking about that. Sarcoplasm is another name for its cytoplasm. Don't let it blow your brain. I told you sarco is a different prefix that we use for muscle. Sarcoplasm, that should be pretty easy. It's going to be, the fluid that will surround all these myofilaments and the nuclei within the cell itself. Are you with me? Okay. Sarcolemma is the plasma membrane. It's just the membrane around it, just like we'd have around a normal cell. Myofilaments are actin and myosin when we're talking about skeletal. I primarily focus on skeletal muscle for this, okay? Smooth and cardiac are slightly different, so we're going to use the skeletal muscle as our composite to understand these parts. Actin and myosin are these tiny little filaments, or myofilaments that we find within that muscle cell itself. And what they do, I like to give you this visual. So actin is the thin, myosin is the thick. So if we look over here, can you see it okay if I leave the lights on? Okay, so here's our thick filament, here's our thin filament. So basically what happens when we shorten a muscle is these little head-like structures which are on the myosin will then attach to the actin. There's little kind of protectors that cover the attachment sites on actin in a wrestling muscle. When we activate it, or we send an action potential to it, those sites are exposed, and this little globular head will attach to the thin filament, which is actin, and move it. Are you with me? And they just slide it. So imagine this is a myosin or a thick filament. This is actin. Those little globular heads, I don't know, this was a good analogy for me. Attach, and what do you think they do? Then they detach and they reattach, shorten it. So if I just keep doing that, I'll shorten that muscle. And if you have millions of these teeny tiny myofilaments doing that, you're going to shorten the full length of the muscle. So once again, skeletal muscle, does that make sense? Skeletal muscles are full of these actin and myosin filaments. Actin has attachment sites on it that get uncovered when the muscle cell is activated. Myosin has these little globular heads which attach to it and shorten the length of the muscle. Are you with me? Does that make sense? Okay. When we stop activating it, those myosin heads will help to put it back into place. Okay, kind of like unhinges to its normal length. Does that make sense? While I'm here and we're talking about this, if we talk about the heart as a little side note, The heart muscle does a similar thing. Attaches, shortens, right? Attaches, shortens. If we get a big flabby heart, what will happen to those attachment sites? Will they overlap as well? If you can imagine this stretching out, what happens? They can't reach as well. That's why when we get a big stretched out flabby heart, it doesn't beat as effectively because you don't have that ability to shorten as well. Does that make sense? I wouldn't test you about that, but I think clinically it's an important thing to know. Okay, so can we picture that? Can we picture the muscle cell, the cytoplasm within it, right? The sarcoplasm, and those myofilaments inside. Are we good with that? Because we're going to move on. Okay. So here's a picture of it. If you've taken muscle physiology somewhere else, they go over all these different parts. A band, I band, Z band, H, I don't. I just don't. I don't know, whatever. I want you to understand how those actin and myosin filaments attach and shorten. That's what I want you to know. Okay, so this picture's up here. You don't need to know all these different subdivisions. I just think it's a good, helpful picture for what we're going to be getting into. Okay? And I like this picture, how you have the sarcolemma, and then you have all these fibrils within the cell itself, just to give you a good visual for that. Let me see. Oh, okay. All right. So, this is how it goes within this muscle cell. How do we stimulate these muscle cells? We send a what? By a nerve. And action potential, right? And action potential comes down that nerve, sends neurotransmitter to the muscle cell, stimulates that muscle cell. Are you with me? Yes? Okay. Now when the action potential goes down the muscle cell, what it does then is it releases calcium into the muscle cells themselves. Those muscle cells allow actin and myosin, those calcium cells allow actin and myosin to join together. Are you with me? So it's the calcium that allows that to happen. You guys are so fried, aren't you? I want to get an espresso machine and bring it in here. Okay, so can you picture that? The nerve talking to the muscle. The muscle has an action potential. That action potential sends calcium in to allow actin and myosin to shorten. Are you with me? Does that make sense? Okay. The way we do that is this netting right here is called the sarcoplasmic reticulum. That's what stores the calcium. When we send an action potential down a muscle to release that calcium, it comes out of this wonky little network or netting of sarcoplasmic reticulum. It goes into that muscle cell, okay? Don't worry about terminal cisternae. Don't worry about that. It's just the ends of the sarcoplasmic reticulum. And then you have what are called T-tubules. And the T-tubules kind of dive down into the cell itself and send the calcium down into it. All right? To help facilitate the contraction of those myofilaments. Okay? So, action potential, right? Comes by a nerve, sends neurotransmitter. I'll show you those steps in just a minute. Action potential goes down the muscle itself, sends a signal to release what? Calcium from the sarcoplasmic reticulum, goes down in those T-tubules into the cell, allows actin and myosin to contract. Yes? Okay. Good. Do you remember the steps of an action potential? What was my definition? Action potentials allow muscles and nerves to work, right? Or function. Excuse me. What we're going to focus on specifically here are motor nerves. So when you're talking about a motor nerve, does anyone remember where the starts in the brain? We talked about it. Pre-central gyrus. Good. Okay, pre-central gyrus. So if I want to move my big toe, I have one nerve come from my pre-central gyrus down to the cord, and I have a second nerve go to my toe. It's a two-nerve complex. That's important to know. Okay? There's some exceptions, but with motor, we have the nerve come from the brain to the opposite side, remember, and I'm talking about where it crosses over later. Maybe I already talked about it. Okay, crosses over. It's called a pyramidal cell. Is that ring a bell? No? All right. Crosses over. I have another nerve, or the second nerve, go to the big toe. Okay? Are you with me with that? So two nerves. That's crazy, right? That's crazy. It's going to control our motions. Okay. It goes to the front of the cord. Do you remember when we talked about the cord? The front of the cord is motor. The back of the cord is what? Sensory. That's something you should always take with you, clinically speaking, right? If somebody has damage to the front of the cord, they'll have motor loss. If they have damage to the back of the cord, they'll have what? Sensory issues. Okay? So this motor nerve is going to go from here to the front of the cord. From there, it'll send a nerve out to my big toe. Okay? I think those are important things to take with you. That nerve then will divide as it enters the muscle. Okay? Each muscle fiber will have one junction with that end of that axon. Okay? Do you remember what that space is called, where the axon talks to the muscle? It's called neuromuscular junction because it's where the nerve talks to the muscle, but the specific name for that space is called a synapse. Okay? Yes, synapse. The entire thing is a synaptic cleft. This space is a synaptic cleft, or this space is synaptic cleft. Okay? So does this look familiar? It should. I'm going to go through this quickly. If you don't remember this, come see me. So this is where the nerve talks to the muscle cell. This is a synapse. Okay? This is a synapse. So we will, let me give you a once-over, have an action potential come down this axon. We release what into this synaptic cleft? Acetylcholine, you could say a neurotransmitter, specifically acetylcholine. Calcium is going to be in these little vesicles here. That'll trigger the release of the acetylcholine. Acetylcholine will attach to the muscle and tell that muscle to perform an action potential. So basically we have action potential, neurotransmitter, action potential. Okay, does that ring a bell? If it doesn't, come see me. I'm happy to go over it with you. I think I have the steps right here. Okay, so synaptic cleft is going to be the small space between the nerve and the muscle. We'll release calcium. That'll trigger the release of acetylcholine into this space. Acetylcholine is going to be the neurotransmitter that we see in the muscular system. That's an important thing to know. Acetylcholine will then trigger the stimulation of the muscle. Okay, and I have the steps right here. So the action potential coming down the nerve will release calcium from little vesicles stored at the end of the nerve. That calcium triggers the release of acetylcholine. Is this ringing a bell, guys? Okay, yes?
So I know the postsynaptic membrane is the membrane on the bottom of the white part. And the presynaptic, is that just the yellow?
The end of the axon.
Okay, because it's not like labeled on anything, so I just want to
Yes,
make sure.
presynaptic membrane, postsynaptic membrane. But that was our composite. We now know it's an axon and a muscle. Do you see what I'm saying? But this is the presynaptic membrane, that's the postsynaptic membrane. Yeah, remember? Okay, acetylcholine will go across, it'll attach to muscle. That muscle then will perform an action potential. Are you with me? Acetylcholine then breaks down either into its subparts or by acetylcholine esterase, which is an enzyme, which will break it down. Any questions on that? No? Yes, no? Okay. Because we're going to kind of go the next step. So we sent the signal from the brain, comes down, talks to the muscle, stimulates the muscle. Okay? In action potential, do you remember all these steps to the action potential? Okay? An action potential is that electrical signal that makes nerves or muscles work. It's got several steps that we can actually chart on a little voltmeter, right? And we can see what's going where. Now, do I have? Do I have a bigger one? I'm going to kind of go through this quickly because I'm hoping to get through a chunk of this lecture. Let's see. But if it doesn't make sense to you, please come back and see me. I'm just going through it quickly because we did it pretty extensively last semester. So if we're talking about a cell at resting and then an action potential, it'll look somewhat like this, okay? We've got a resting cell we'll measure. So if I took two little voltmeters and stuck it into that cell, the membrane of that cell would be negative what? 70, right? An action potential will stimulate that nerve or that muscle to function. Okay, the numbers might be a little bit different, but I'm going to go over the same numbers that we went over last time. So when we stimulate that muscle or nerve, that stimulation will look like this. And the reason we get this chart is because we have an influx and an outflux of what? Different, what are they called? Those little charged particles. Ions. Are you with me? Okay. Do you guys remember this or no? We do. Okay. So, muscle's just hanging out, chilling, just doing whatever it wants to do. We stimulate it. This is the electrical recording of what it will be doing. The first step is, wait, let me go back. Sorry. Wrong way. Depolarization. Okay, so back to this chart. Depolarization. So we open up voltage channels in the cell that allow what to come in? Positive sodium. And if it's positive, it'll make the chart go up because we have sodium moving into the cell, making it more positive. Are you with me? If we have enough sodium come in to make it to negative 50, do I have this up there? It's not on there. It's on the next one. Negative 50 or 55, I'll take either one, is threshold. If you make it to negative 50 or 55, you're going to have an action potential. Okay? Are you guys with me? Right? That action potential then will peak all the way up to positive 30, right? Okay? At that point, sodium channels close. What channels open? Potassium channels open. We undergo repolarization. Potassium is going to move from inside to outside of the cell. If we pump a bunch of positive ions out, that cell will become more negative. Okay, are you with me? Eventually we get back to negative 70, but potassium channels are slow to close. And we have what's called hyperpolarization. Is that on there? Hyperpolarization. I don't think it's on the next one. Okay. Does this ring a bell? Okay. Basically what you need to know is an action potential makes a nerve or muscle work. There's multiple steps to it. These are the steps, okay? Depolarization is when sodium channels open. Sodium enters the cell, making it more positive. Once it reaches its peak of positive 30, they will shut off. And then potassium channels open, right? Potassium will move from inside to outside of the cell, making it more negative. Until it reaches negative 70, they're slow to close, so there'll be a little swoop down of hyperpolarization.
If it doesn't reach positive 30, it's not an action potential.
If it reaches negative 50, it's going to reach positive 30. But there's variations to that. What we're going with is this, because there's different numbers with different cells. So, okay. Yes? If this is not seeming familiar, come see me, okay? By the way, I'm not going to be here tomorrow. I drove all the way out to Rhode Island and back last week, and I'm behind. I got to catch up on work. But I'll be here Thursday for sure, if anybody needs me, okay? All right, so refractory period is just when you can't stimulate another action potential, okay? It's got to reset itself in order to stimulate another action potential. Yes, no? All right, come see me if it doesn't make sense. I'm more than happy to help. Here's a little snapshot, okay, to give you an idea of how that goes. I also really like the lecture from Neuro. I go more and more into it in neurology, so you can always go back and look that up on YouTube. Okay, so an action potential will go down the length of that muscle cell, okay? It'll stimulate the release of calcium into that muscle cell. That calcium unlocks those binding sites on actin. Myosin can attach then, and what happens to the muscle? Shortens. Okay? You're done with your contraction. Calcium will release. It'll unbind itself, lengthen the muscle, and go back to resting and reset everything. Calcium will go back into the storage tanks for the sarcoplasmic reticulum. Okay? Does that make sense? That's a lot. Okay? Come see me if it doesn't. Muscles respond to stronger stimuli, so you can recruit more muscle fibers or you can do multiple stimulations. You'll see in your book it goes way more into it than I go into it. I kind of keep it a little brief. Okay, that's better. Okay, so you can recruit more muscle fibers or you can have more signals that go to a muscle repeatedly. Okay. And obviously you'd release more calcium if you were doing that. Now, muscle tone. Let me see. It's almost 10 after, right? Let me see. How many more slides do I have? I'm going to finish this because we only have like 7 slides left. You can absolutely leave if you want to. I'm going to finish this so it's done for the video. Whatever you want to do. I don't think it's going to take me much more than 5 minutes. Okay, so if you have to go, by all means, go ahead. Muscle tone, I taught you about, really, if you have to go, I won't, it's fine. Okay, muscle tone is when I taught you about the muscles having that slight contraction. And I think the 2 good examples that we should know is the, I'll give you no if you need one. No, just kidding, right? Your postural muscles, but also the tonicity of the GI tract, which we'll talk about when we get into the GI tract. The GI tract's always slightly contracted, so if you were to dissect a cadaver, it would be much longer than it would be in us, because it's relaxed. Okay, so tonicity. Isotonic versus isometric. So isotonic movement of a muscle is moving it through space against resistance. So when I walk over and pick up my tea, I'm actually moving that muscle. But if I were to walk over and miss my tea and grab this counter and lift, is this muscle working? Is my biceps working? Absolutely. Is it moving? That's the difference. With isotonic, the length of the muscle changes. With isometric, it doesn't. You have resistance or movement, or I shouldn't say movement in a muscle. You have contraction of a muscle without movement. This is used quite often when you talk about rehab for people. If someone's undergone a very difficult injury, say in their neck, you don't want to go swinging their head around. You'll undergo isometric strengthening first, where you push up against the head but not go through a big range of motion. Okay? So tonicity, isotonic versus isometric. Okay? Muscle metabolism. 3 ways we could get muscle. Okay, now once again, we're going to take glucose and convert it into ATP. I have an entire lecture, not an entire lecture, but a large lecture that will show you all these steps. So realize this might not make complete sense at this point, but we'll go into this in depth in the future. We can directly phosphorylate ADP into ATP. That simply means you add a phosphate to it. Okay, we have free phosphate floating around in our body. We can take it and add it to an ADP and make it ATP, and then we have that stored energy. Okay. Anaerobic mechanisms, which will convert lactic acid, I'm going to talk to you about this in the future, okay, into ATP, we get 2 ATP. But the big one, the big money maker, guys, is really going to be aerobic respiration. This is a whole lecture. I'll get into this. But basically what you need to know is if we have glucose and we have oxygen, we can make a whole bunch of ATP. We can make 36 out of that process. It's called the Krebs cycle or citric acid cycle. I'm going to get into that in the future. I just want you to know that you need glucose and oxygen. Are you guys breaking out in hives? Talking about the Krebs cycle? Okay, good. To create that energy. Okay. Muscle fatigue will happen when glycogen and oxygen run out. Obviously, you don't have energy to make those muscles contract. We have slow fibers, we have fast fibers, and we have a mix of them. That's why some people are really good at sprinting, and some people are really good at heavy weightlifting. It's a mix of these slow versus fast fibers. Okay, and their descriptions are right there. I'll let you read through those on your own. Okay. Microscopic structure of smooth muscle. We know it's spindle shape. I showed you how it's those long linear ones, right? When we talk about the GI tract, and I will show this to you in the GI tract, you have a longitudinal and a circular layer of that smooth muscle. And the reason that we have a circular layer, so what that means is the cells, if you imagine my fingers being the cell around the tube of the GI tract, the circular layer will have the cells going in a circle. Longitudinal will have them going lengthwise. That gives us the ability to churn our food. When we were cavemen, we didn't get a lot of food. So our bodies were built over millions of years to suck all the nutrients out of our foodstuffs. So that longitudinal circular layer in the GI tract churn your food so we can get as many nutrients as possible out of it. Okay? Does that make sense? Sort of, kind of? Okay, I'll go over that in the future. Contraction of smooth muscle, it has actin-myosin, is triggered by energy with ATP. Low energy requirements because its contractions are slow and steady. You don't want your GI tract going fast, trust me. Okay, that's going to create problems possibly both ways, if you get what I'm saying. Our GI tract is meant to move slowly, okay, and churn that food stuff. That's a visual that nobody wants, right? So neural stimulation of muscles. So we know that we have acetylcholine for skeletal muscle. When we're talking about smooth and cardiac, you can have acetylcholine or norepinephrine. To remember parasympathetic and sympathetic innervation. Parasympathetic calms us down. That will release acetylcholine. Sympathetic does what? Fires us up. That releases norepinephrine as its neurotransmitter. I think that is important to know. So parasympathetic acetylcholine, sympathetic norepinephrine. Okay? Now each has different effects depending upon the receptors. In general, acetylcholine will calm, norepinephrine will increase, but that really gets a little wonky when you talk about receptors. Okay. We also have different hormones which can affect muscles. We'll talk about that in the future. Okay? Muscular dystrophy is a muscle destroying disease. Fat and connective tissue can actually get deposited into your muscles. If you do that, those muscles don't work efficiently, right? Because they don't have muscle cells where the connective tissue and the fat is. There's different degrees of it. You can have a very mild form. You can have a very advanced form. Let me tell you the stereotypical sign with children. If you have a child that doesn't just hop right up and start playing, if when they start to get up, they walk themselves up like that, that's a very stereotypical sign of muscular dystrophy in a child that needs to be checked. okay? Myasthenia gravis is an autoimmune disease that destroys those acetylcholine receptors at the mortal end plate. So if you don't have the proper amount of acetylcholine receptors, you can't activate your muscles as well. These patients, if they're given it extra acetylcholine, are active because it can stimulate more of those receptors that they still have, okay? But without it, they have no energy. All right, we made it.